Now, the question that took us to this compound page was, how does E. coli get its arginine? So, one way that any cell can get arginine is to pick it up exogenously, pick it up from the environment. And to do that, we're going to expect there to be a transport reaction, and if there is one, we know it's going to be linked off this page. And actually, we have two transport reactions. We have this transport reaction, and we have this one. And I'm going to go with the second transport reaction, which is ATP-dependent transport of L-arginine from the periplasmic space into the cytosol. And so I'll just click on that and end up on a transport reaction page. Now, a transport reaction page is a special type of reaction page. We'll be looking at what you might think of as more typical reaction pages, for example for metabolic reactions, a little bit later on in this introductory series. For the moment, we're going to look at a transport page. Transport page has features that a lot of other reaction pages do. It shows the classification system up top for our reaction classification. In the case of transport reactions, a lot of them aren't populated in the EC system, so we don't have EC numbers for them. Instead, we have our own classification system that says, hey, this is, ATP-driven transporter. We have a protein complex that is actually there are two different transporters here, and we have both of them listed, and you can click through. That would take you to the complex. And we have the constituent parts of each of these complexes listed here. In addition, we have this schematic that shows the transport reaction itself. We have, and obviously this is a very schematic representation, it's just meant to give you the basic impression that arginine comes from outside in the periplasmic space into the cytosol. It uses ATP that's inside the cytosol plus water, and in an ATP-dependent manner transports arginine actively into the cytosol. Now you'll notice I'm mousing over all these things, and it actually brings up these little windows. These are called tooltips, and they tell you what you're looking at. And obviously there's not a lot of extra information you're getting here. ADP, ADP, phosphate, phosphate. It matters more when there are longer names, when it's a more complex reaction you're looking at. But the other thing it indicates is whenever one of these little tooltips pops up, it means I can click on that and it will actually take me to the thing I'm mousing over. So once again, remember our cardinal rule is, when in doubt, give it a click. It might work. So down below that schematic representation of the reaction, and I'll just pull the page down, we have this thing, which is a gene reaction schematic. Now, I'm not going to discuss what a gene reaction schematic is in detail right now, but very briefly, it shows how reactions, proteins, and genes interconnect. And again, when in doubt, and when you don't know what's going on, if you see a little question mark, it means we think you might be confused by that. Click on that, it'll take you to an explanation. This webinar series will address the gene reaction schematic later, toward the end of the webinar series. We're not going to worry about the gene reaction schematic right now. Instead, I'm going to page back up, I'm going to mouse over L-arginine, and I'm going to click on it. And we're back at the L-arginine page. And we've answered one question about how E. coli gets its arginine. We've shown that there are actually two transport reactions it can use, and we've clicked through and we've taken a look at one of them. Of course, picking up an exogenous small molecule is only one way a cell can acquire it. Another way is biosynthesis, and we're interested in arginine biosynthesis, especially since it's going to serve as our example for the rest of this webinar series. So, I will page down, and here we have a link to a pathway reaction that has arginine as a product. That sounds promising, as does the name Arginine Biosynthesis. And I'll click through to this and give you a little taste of a biosynthetic pathway page. This is what we'll be covering in part 3 of the Introduction to BioCyc webinar series. We're going to show you BioCyc pathway pages, we'll show you the kind of information they contain, and we'll show you what you can do with that information using the pages. For now, I'm going to thank you for listening to this part 2 of the Introduction to BioCyc webinar series, and invite you to continue with the series. In part 3, as a reminder, we'll be learning about pathways. In part 4, we'll address genes, the genome browser, and transcription units. And in part 5, we'll look at proteins, protein complexes, and reactions. So we'll look forward to seeing you right back here for part 3.